So the next category is body organization. So based on the body organization, that is the characters such as whether it is having a cellular organization or the tissue organization or the organ system. I hope you are clear with this. So what is the cellular organization? Each and every organism, the prime constituent is cells. So all are made up of cells. And then in the case of Monera, it's cellular organization. That is the independent cells. They do the function. And then in the case of fungi, it is the tissue level of organization. So what is the tissue? These similar cells, they join to form tissues. And then two or more tissues, they join to form organs. And then organ system, these organs, they coordinate and form organ system. So I hope you know this level of organization. So it differs from Monera to Animalia. So in the case of Monera, it is cellular organization, and Protista also it is a cellular organization. And the case of fungi, it is multicellular, and these cells, they form similar cells, they form tissues. And here in the case of plant, it may be either a tissue or the organ level of organization. And then in the case of Animalia, it is the organ system level of organization. And then it also differs according to the mode of nutrition. So what do you mean by this mode of nutrition? It may be an autotroph or a heterotrophic. So what do you mean by autotroph and a heterotroph? So auto means they can synthesize their own food. So how they can synthesize their own food? It can be either they get their energy from light or from the chemical substances. So if it is from light, then it is called the photosynthetic autotrophs. And if they get the source from chemicals, it is called chemosynthetic autotrophs. So the mode of nutrition in the case of Monera is either they are photosynthetic autotrophs or chemosynthetic autotrophs. And heterotrophic mode of nutrition. I hope you are clear with these terms, right? Saprophytes and parasite. So what are saprophytes and what are parasites? Parasites are the organisms which are depending on the living organism. That is, they feed on the living organisms. And in the case of saprophytes, they get their food from the dead organisms. So there is a difference between saprophyte and parasite. So Monera, the mode of nutrition is autotrophic and heterotrophic. It may be photosynthetic autotrophs or the chemosynthetic autotrophs, or it may be heterotrophic, and whether it may be parasitic or the saprophytic. And in the case of Protista, so how the mode of nutrition is? Here also autotrophic and heterotrophic mode of nutrition is present. In the case of fungi, it is heterotrophic only. So they cannot prepare their own food, but they are dependent only on the other organisms for its food. So fungi is heterotrophic. And plantae, you know, it is autotrophic, photosynthetic, and they can prepare their own food. In the case of Animalia, it is heterotrophic. They cannot prepare their own food. And the mode of nutrition is holozoic, that is ingestion. They will have the other food materials and the saprophytic. So the animals are having holozoic and saprophytic and other modes of heterotrophic nutrition. So on the basis of these characteristics, that is cell type, cell wall, and the body organization and mode of nutrition, the animals are classified into 5 kingdoms. So hope you're clear with this classification. So we'll then see about the different types. For example, if you take the 5 kingdom classification, the lowermost one is the Monera. So here it will be Monera. Then there comes the Protista. Then there comes the Kingdom Plantae and Kingdom Fungi and Kingdom Animalia. So on what basis we are differentiating this? That is, here the Monera is differentiated from other kingdoms, like here it is a prokaryote. All the other above Monera, all are eukaryotes. So based on the complexity of the cell. So the in-between Protista, it acts as a link between Monera and other eukaryotes. So here, what is the main difference between the Protista and other kingdoms? For example, it is the complexity of organism. So here, the main difference is all are unicellular. So all these organisms which are present in the Monera and Protista, here they are unicellular, and here all multicellular. And these three, that is Plantae, Fungi and Animalia, here they are differentiated on the basis of mode of nutrition and also on the basis, here actually it is evolving, that is there is an evolution of organisms. And also the ecological factors. For example, we can say ecology means that is Plantae or producers. And here we can say Animalia as consumers and fungi as decomposers. So here also on the basis of nutrition, here it is phototrophic. It is photoautotrophic, and here it is heterotrophic. And here also it is heterotrophic. So this you can easily remember. So the primitive organisms are all Monera, then there comes the Protista, and from the Protista, they are the little advanced organisms, that is the multicellular organisms. So based on the complexity of the cell, complexity of the organism, mode of nutrition and other ecological factors, they help in the differentiation of these living organisms into 5 kingdoms. Then there occurs in domains. So above the kingdom, there are domains. So actually differentiated into 3 domains. This is one type of classification. For example, all the eukaryotes, they are placed under one domain. Then bacteria, and here Archaebacteria. That is, when you compare the bacteria and the Archaebacteria, there occurs a huge difference. So they differentiated the Monera again into 2 kingdoms based on the rRNA studies. They are differentiated into Archaebacteria, then this bacteria that forms the Monera, and then all the eukaryotes are placed under the same kingdom, that is, it includes Protista, then fungi, Plantae and Animalia. So depending on the characteristics. So this is one type of classification, that is domain. The kingdom Monera is divided into, again, two kingdoms, Archaebacteria and Monera, and then eukaryotes as Protista, fungi, plantae, and Animalia. So this is called the domain theory of classification. So next we'll see in the, so I think you are clear with the classification. So first, genuine scientists who first started classifying the living organism is Aristotle. Then there comes the two kingdom classification by Carolus Linnaeus, but there are so many drawbacks in the two kingdom classification. So Whittaker classified the organisms into 5 kingdom, so 5 kingdom concepts, and also there comes the domain of classification of organisms. So then we'll see in detail about the kingdom Monera. So kingdom Monera, we saw that all the prokaryotes are included under the kingdom Monera. But then the domains, only the 5 kingdom classification is accepted. So the Whittaker's 5 kingdom classification is accepted. So first kingdom, this Monera, it also includes the Archaebacteria and other types of bacteria. So this bacteria, they are the most abundant microorganisms. So they are most, that is the, what to say, higher in number. So they are the most abundant microorganisms, and hundreds of bacteria are present in a handful of soil because they are so microscopic and you won't be able to see the microorganisms, but their number is very huge. And they also live in extreme habitats such as hot springs. So they exist in all types of temperature at an extremely high temperature in hot spring, even up to 100 degrees centigrades, and also in deserts, snow, and deep oceans. So all types of environment, they are able to survive and they are able to adapt to that particular environmental conditions. And many are parasites. So what do you mean by parasite? I already saw they are dependent on the other living organisms. So they are parasites. So based on the shape, there are different types of bacteria. The first one is coccus. The coccus is spherical in shape. The spherical shaped bacteria are called coccus, and rod shaped bacteria are called bacillus, and comma shaped bacteria are called vibrio, and spiral shaped bacteria. So these are the different types. Cocci is spherical, bacillus rod shaped, spirilla, and then comma shape. So these are the examples for the different types of organisms. So I think you are more familiar with these bacteria like Streptococcus pneumoniae, and then so coccus, they are spherical, and then Staphylococcus. These are some of the examples of the bacteria. Staphylococcus aureus, then Clostridium. So here they are all rod shaped, and this is a spiral example is Troponema pallidum. So these are some of the examples of the bacteria with different shapes. That is coccus, bacillus, vibrio, and spirillum. So if you see the structure of bacteria, so what are the differences between this bacterial structure and other eukaryotic cell structure? For example, here the bacterial structure is very simple, but they are complex in behavior. But since it is a unicellular, so all the characters, that is all the functions are done by a single cell. So complex in behavior, and they show metabolic diversity. And some bacteria are autotrophic. So you know, autotrophic means they can synthesize their own food. So how they are synthesizing their own food? From inorganic substrates. So they are synthesizing food from inorganic substrates, and majority are heterotrophs. Some are autotrophs, but majority are heterotrophs. That is, they do not synthesize their food, but depend on other organisms or a dead organic matter. So if they're depending on dead organic matter, they are called saprophytes. Called saprophytes. So if you see the structure of bacteria, so what are the features that you can see? Outer is the capsule, and then comes the cell wall, which is present in the prokaryotic cell. And the characteristic feature of this bacterial cell wall is it contains the compound that is peptidoglycan. But in the case of plants, the cell wall is made up of cellulose. So here it is made up of peptidoglycan, then plasma membrane, which is common in both prokaryotes and eukaryotes. And then it has a cytoplasm, ribosomes are present. So we'll see in detail about all these organelles that are present in the chapter that is cells. So I hope you know the basic structure of a prokaryotic cell, and it contains plasmid, which is present only in a prokaryotic cell. So plasmid, what are plasmids? They are nothing but the extra cellular, that is genetic material. This is the nucleoid. Prokaryotes won't have a proper nucleus, but they have a structures that is called the nucleoid. That is, they don't have the proper nuclear membrane. No nuclear membrane. And there are no membrane-bound organelles in the case of a prokaryotic cell. And plasmid is one of the characteristic feature in a prokaryotic cell. That is, it is an extra chromosomal genetic material. And also they have these structures called pili, which is used for attachment, and flagellum. So you can see in detail about all these structures in that cell chapter. So this is the general structure of bacteria. Then there comes the classification. So how are the bacteria are classified? These bacteria, first category is the Archaebacteria and Eubacteria. So what do you mean by Archaebacteria? These Archaebacteria, they exist in a extreme conditions. That is, they can tolerate the wide range of conditions. For example, halophiles. So halo means salt. They can exist in high salt concentration. And then thermoacidophils. So what are thermoacidophils? Thermo means high temperature. So they can exist commonly at 80 degrees centigrade and even tolerate up to 100 degrees centigrade. Then acidic conditions, for example, at the pH 2. So they can exist at the pH 2. They are thermoacidophils. And then methanogens. What are methanogens? That is, they can form methane. So methanogens are generally present in the ruminants. So they can form the methane. And then eubacteria. Under eubacteria, autotrophs, they can synthesize their own food. Photosynthetic autotroph, photo means light. Here they can synthesize their food. Then photosynthetic means with the help of light, and chemosynthetic means with the help of chemicals. And the next classification is heterotrophs. So this already we saw, either it may be parasitic or saprophytic. So these are the different types of classification of bacteria, that is Archaebacteria and Eubacteria. So this autotrophs and heterotrophs are on the basis of mode of nutrition. And then depending on respiration, again, they can be classified either into aerobic and anaerobic. So from the term itself, this requires oxygen. Here they can exist in the absence of oxygen. And under this, we can say it is obligate or facultative. And under anaerobic, it is obligate or facultative. So what do you mean by this? Obligate means they are strict aerobes. That is, they only in the presence of oxygen they can survive. And obligate anaerobes means only in the absence of oxygen they can survive. And facultative aerobes and facultative anaerobes. That is, though they need oxygen, and some organisms they can survive even when oxygen is not there. So facultative means they will adapt to that particular conditions. So that is the difference between the obligate and facultative. So on the basis of respiration, the bacteria are classified into aerobic bacteria or anaerobic bacteria. So hope you are clear with this till now. That is about the types of classification and about the structure of a bacterial cell and about the types of bacteria, that is Archaebacteria and Eubacteria. So next we'll see about the Archaebacteria. Yeah. So this Archaebacteria, actually, they are the most primitive organisms. That is the most ancient organisms. So we can call this Archaebacteria as the living fossils. Call them as living fossils. So what do you mean by this living fossils? That is oldest living fossils. So these organisms, they are the first organism. They are believed to be the first organism to be existing on this earth. So they believe that because of that different cell structure, they were able to tolerate the wide range of conditions that were prevalent in those days. So during the origin of, that is the first occurrence of living organisms, we don't even know how the environmental conditions would have been. For example, there may be in high temperature or the different types of extreme conditions of the chemicals. So all those things were tolerated by this Archaebacteria because of their different cell structure. That is the cell structure, they contain complex polypeptides and polysaccharides. So they have a different cell structure that help for the survival in extreme conditions. So this is one of the main factor that help them to survive the harsh conditions. So they live in harshest habitats such as extreme salty areas like halophils. There halo means salt. Phils mean generally loving. So they exist, those type of bacteria which exist in high salt concentrations. Then hot springs, that is thermoacidophils, and marshy areas, they have more amount of methanogens. So these Archaebacteria are called ancient living fossils, and they have the different types of cell structures that differentiate the Archaebacteria from other Eubacteria. That's why they thought of putting it into a different kingdom in that domain area of classification. So they have a different cell wall structure for the survival in extreme conditions. And then methanogens. So what are methanogens? They are present in the guts of ruminant animals like cows, buffaloes, etc. And they produce methane gas. So they produce methane from carbon dioxide from the dung of these animals. They are present in the guts of ruminant animals, so they also help in the digestion of cellulose. Know that they are all herbivorous animals, so they eat plants, help in the digestion of cellulose, since they are present in the guts of the ruminant animals, and they produce methane gas from the dung of these animals. So these methanogens are also one of the Archaebacteria. So Archaebacteria includes halophils, thermosidophils, and methanogens. So are you clear with this? Archaebacteria. So then we'll move on to eubacteria. Yes. So what are eubacteria? So they are called true bacteria. Eubacteria are called true bacteria, and they have a rigid cell wall and a flagellum if motile. So in the case of motile organisms, the movement is with the help of flagellum. These includes autotrophs and heterotrophs. Eubacteria includes both autotrophs and heterotrophs. So the first category is the photosynthetic autotrophs. Example is cyanobacteria. Cyanobacteria is also called blue-green algae. So you would have heard about cyanobacteria. So they are similar to plants in that they are photosynthetic. So the cyanobacteria is similar to plants in that they are able to photosynthesize. So what is the reason for it? Because they contain the pigment chlorophyll A, similar to that of green plants. So they can photosynthesize and they can produce their own food. And then cyanobacteria. They are unicellular, single cell, and they exist in colonies, colonial or filamentous structures. They are marine or terrestrial. They live in oceans, and also some of the cyanobacteria are terrestrial. And these colonies are generally surrounded by gelatinous sheath. So this is the mucilaginous sheath. That is the outer, the blue one is the mucilaginous sheath. And they have the special structures called heteroses. So these are the heteroses that are present in the cyanobacteria. And the role of this heteroses is they help in fixing the atmospheric nitrogen. Fixing of atmospheric nitrogen. And one of the main harmful thing about this cyanobacteria is they often form blooms in polluted water bodies. So there will be overgrowth of this cyanobacteria, and they form the hazardous thing in the polluted water bodies. So this is one of the main hazard of the cyanobacteria. So they give the very bad odor to the water. Because of that, more formation of this cyanobacteria. They form in colonies on the surface of water. This is about the cyanobacteria, which is an example for a photosynthetic autotroph. So next is the hemosynthetic autotrophs. So what are hemosynthetic autotrophs? From the name itself, we can see. So by seeing the name, hemo means chemicals. Synthetic means they can synthesize from the chemical compounds. So autotrophs are, they produce their own food. So they oxidize the inorganic substance such as nitrates, nitrites, and ammonia, and they use the released energy for ATP production. So instead of light, they are using the inorganic substances. What is the main use of this? They help in recycling of nutrients. So wherever there are more accumulation of nitrates or nitrates, this bacteria, there is this category that is hemosynthetic autotrophs. They help in the recycling of nutrients, the utilization of these chemicals, nitrogen, phosphorus, iron, and sulfur. So they help in degradation of these substances, that is recycling nutrients. And then as the heterotrophic bacteria. So what are heterotrophic bacteria? The first two categories, that is autotrophs. In autotrophs, we saw the photosynthetic autotrophs and chemosynthetic autotrophs. And the case of heterotrophic bacteria, they cannot synthesize their own food and they are dependent only on the other organisms for its food. They are most abundant in nature. So when compared to autotrophs, these heterotrophs are more abundant. The majority are important decomposers. So they help in the decomposition. So they depend on the dead organic matter. So they help in decomposition of the dead organic matter. These are the different types of eubacteria. That is photosynthetic autotrophs, then chemosynthetic autotrophs and heterotrophic bacteria. So I hope you are clear with this. Then there are several types of bacteria which help the human. There is impact on human affairs. There are also positive as well as the negative impact. For example, if you take the formation of curd from it. So what is the bacteria that is involved in the formation of curd? So daily you would have seen in your house the formation of curd. So how the curd is formed? Because of the action of bacteria. So main bacteria which is involved in the formation of curd is the lactobacillus. So bacillus is an example for a rod-shaped bacteria. This lactobacillus help in the formation of curd from milk and also in the production of antibiotics. Example, streptomycin is an antibiotic which is produced with the help of bacteria, and the main organism which is involved is Streptomyces gracious. And then fixing of nitrogen in legume roots. The example is rhizobium. And there are some pathogens which cause diseases like cholera, which is caused by Vibrio cholerae. And then typhoid, which is caused by the bacteria that is called Salmonella typhi. And then tetanus is caused by Clostridium tetani. So there are different organisms which cause diseases, and citrus canker. It is one of the diseases which is caused or which affects the citrus plants. So it affects both plants as well as the animals. So these heterotrophic bacteria, they have various impact on human affairs. So next is the reproduction. So how these bacteria reproduce? They reproduce mainly by fission. So what do you mean by fission? For example, if it is a bacteria, the nucleus, then other organelles will be there. Then it grows first. Then there occurs a division. There will be a formation of arrow here. Here is the nucleus, and then it divides into two organisms. Nucleoid. So bacteria reproduce mainly by fission, but under unfavorable condition, they produce spores. So the spores are called endospores. Oh, what are endospores? So these spores, actually they are in the dormant form, and they're able to resist the different type of conditions, environmental conditions like temperature or pH. So they are the dormant forms, and they help the survival of the bacteria. So under favorable conditions, they produce spores, particularly endospores. They also reproduce by a sort of sexual reproduction. That is, the DNA is transferred from one bacterium to the other by means of sexual reproduction. So sexual reproduction means generally it involves the two parents. Here how the transfer is happening is there are so many modes like transformation, transduction. So that we'll see in detail under the cells chapter about the bacteria. Then the next type of bacteria that comes under the Kingdom Monera are MYCOPLASMAS. So these mycoplasmas are the organisms without a cell wall. There is no cell wall. So all the bacteria have a cell wall which is made up of peptidoglycan, and here there is no cell wall. And they are the smallest living cells known. These mycoplasmas are the smallest cell and they can survive without oxygen, anaerobic conditions, and many are pathogenic in animals and plants. So they are pathogenic. These are about the mycoplasmas. So under bacteria, we saw about Archaebacteria, eubacteria, and then mycoplasmas. So under eubacteria, photosynthetic autotrophs, chemosynthetic autotrophs, and heterotrophic bacteria. And among the heterotrophic bacteria, they have many roles like the formation of curd, and also they have many other roles like these bacteria in industries, like formation of vinegar, and also the bacteria that is E. coli. It is one of the pollution indicator. That is, if more amount of bacteria is present in the water, then we can say that there are more amount of pollutants. So it is a pollution indicator, and also the Pseudomonas putida, that is involved in the degradation of oil. There are many other commercial or the industrial applications of the bacteria. They play a diversified role. So I hope you are clear with this, and any doubts, maybe we'll discuss few questions related to this, that is classification and the Kingdom Monera. So which one of the following are comma shaped? Is it coccus, vibrio, bacillus, and clostridium? Yeah, it is option B, vibrio. Yes, good, Nandini. So it's not coccus. Coccus is spherical. This is rod shaped, and it is not clostridium. Yeah, good. Then we'll go to the next question. Most of the bacteria are. So what is the answer? Whether it is an autotroph, chemoautotrophs, or heterotrophs and parasites. Yeah, option C. Most of are heterotrophs. That is, they can't make their own food. So option C is the correct answer. They are heterotrophs, not chemoautotrophs. Only few are autotrophs. So mostly they are heterotrophs. So option C is the correct answer. Yeah, good, Nandini. Then the next question is, which of the following statements is wrong about Archaebacteria? Whether they live in some of the most harsh habitat, they are recently evolved group, cell wall is peptidoglycanless, they are unlike most other bacteria. So which is the correct option in this? The third question, option B. Yeah, very good. So I hope you're clear with the concept. So they are recently evolved group. That is the wrong answer. So you have to be clear whether it is given as wrong or correct. So the wrong answer is They are recently, because we know that Archaebacteria are the ancient living fossils. And peptidoglycan less. So they differ from the eubacteria in their different cell structure. So they don't have the peptidoglycan, and they are not like other bacteria, and they live in marsh habitats. So the correct option is section B. So the fourth question is, which of the following areas or conditions would be favored by thermoacidophils? Thermoacidophils. So thermo means high temperature. Acidophils mean in the acidic conditions. So which one? Whether it is the stomach of many herbivores, hot alkaline, hot sulfur springs, and deep sea volcanic. So which will be the correct answer? Fourth one. So option B is, option C is the correct answer. It's not option B, Sophia. So for example, option B, it is written as hot alkaline. Alkaline means, so here it is acidophil, that is, they will exist only in the acidic conditions and not in the alkaline conditions. But here it is hot alkaline, and in the case of option C, it is hot sulfur springs. So the sulfur springs only, they provide the acidic conditions which are required for the growth of this thermoacidophils. So this is the acidic environment. So C is the correct answer. So are you clear with that now? It's not option B, it is option C. So, and then the fifth question is, which is not correct about methanogens? They are Archaebacteria. They live in marshy areas. Methane is their preferred carbon source. They are present in guts of several ruminant animals, cow, buffaloes, and they produce biogas from the dung of these animals. So which is the correct option? Yes, option C is correct. Why it is option C? You able to follow this then? Methanogens, methane is the preferred carbon source. So actually what happens? They form the methane. So that is not the preferred carbon source. So this is the incorrect option. The other options are all correct. They are Archaebacteria. They live in marshy areas. They are present in guts of several ruminant animals, and they produce biogas. So the next question, some of the cyanobacteria can fix atmospheric nitrogen in specialized cells called, I hope this question is easy for you, so you can easily say the answer. Sixth question, akinets, heterocyst, homocyst, and nodule. Yeah, it is option B. B is correct. So you check the options clearly actually. So this is heterocyst and homocyst. So maybe you would have got confused by these terms, hetero and homocyst. So the correct option is option B, heterocyst, or the cells that can fix atmospheric nitrogen. So heterocyst, it's not homocyst or the akinets. Then the next question is, the group of organisms oxidizing various inorganic substances such as nitrates, nitrites, and ammonia using the released energy for the ATP production are heterotropic bacteria, Chemosynthetic autotrophic bacteria, photoautotrophic bacteria, saprophytic bacteria. So which is the correct option for this? For the 7th question, the correct option is option B. Yeah, I think you all are clear about it. Good, Nandini and Sophia Beham. So 7th, the option is option B. Chemosynthetic, because they can produce their own food with the help of chemicals. Then all of the following diseases are caused by bacteria except, I hope it is an easy question so everybody can answer. Yes, good, Nandini, it is option A. The other diseases are all bacterial diseases, cholera, tetanus, and typhoid, but flu is caused by a virus. So A is the correct option. Yeah, good. So the next question is, bacteria reproduce mainly by their endospore formation, zoospore formation, fission, or sexual method. Though we saw about the all modes of reproduction, they asked mainly by. So you read the question properly, then answer. Right. So the answer, it's option C. Yeah, good, Nandini. It's by fission. Okay, so bacteria, the reproduction is mainly by fission. So, you know, endospores and sexual method also exist, and zoospores are the spores that are motile, that is the for mobility. Fission. Yeah, good. Yeah, Beham. So which of the following play a great role in recycling nutrients like nitrogen, phosphorus, and sulfur? For the recycling, that is, they use that nitrogen, phosphorus, and sulfur. So what type of bacteria? Whether it is chemosynthetic autotrophic bacteria, parasitic bacteria, photoautotrophic bacteria, or cyanobacteria. Yes, it is option A actually. Yeah, correct, Christopher. So why it is option A? Because this recycling of nutrients, that is, they have to utilize the nitrogen, phosphorus, and sulfur. Nitrogen, phosphorus, and sulfur. What happens in the answer is chemosynthetic, because they use all the chemicals which contain, for example, nitrates that we saw, right? Nitrates, nitrites. So wherever there is an accumulation of all these chemicals with the form of nitrogen, phosphorus, and sulfur, these bacteria, they use these chemicals for the production of food. So they help in the recycling of nutrients. So you say the term recycling. So it is mainly done by chemosynthetic autotrophic bacteria. A is the correct answer. And what these parasitic bacteria means, they are dependent on the living organisms. So it is not the option. And photoautotrophic means they use the sunlight. So there is no rule of recycling of nutrients. And also the cyanobacteria is also what type of bacteria? It is also a photosynthetic bacteria. So the option is chemosynthetic autotrophic bacteria. Okay. Yeah, so which one you want to explain? This question? Are you clear now, or you want me to explain again? 10th question. Okay. The question is, which of the following play a great role in recycling nutrients like nitrogen, phosphorus, and sulfur? So the option is chemosynthetic autotrophic bacteria. What do you mean by autotrophic? That is, they can produce their own food. Autotrophs are the organisms which can produce their own food. So what happens in the case of chemosynthetic autotrophic? So they produce their food from these chemicals. that is nitrogen, phosphorus, and sulfur, they use this chemical. So they help in the recycling of these chemicals, and they help to form the food out of these chemicals. So there occurs a recycling of nutrients from these chemicals. So the option is chemosynthetic autotrophic bacteria. So this is the correct option. So are you clear now? Okay. Then we'll go to the next question. Match the organisms in column 1 with habitats in column 2. So how will you do this? First, halophils. Can you try this? Halophils are the bacteria which exist in the salty areas. So what will be the thing? A. Yeah, so what will be the answer for that halophils? A is salty areas, that is 4, and then B will be thermoacidophils, that is they exist in hot springs. So the option will be for B, it is 1, and methanogens, they live in the guts of ruminants, and then D, cyanobacteria, they live in the aquatic environment. So this correlates with option B. So B is the correct answer for this. So are you clear with this? So any doubts in this? So I hope you are clear with the thing. That is the classification, introduction about the 5 kingdom classification, and about the Kingdom Monera, that is about the types of bacteria, that is Archaebacteria, Eubacteria, and Mycoplasmas. And it's all about the different properties or the characters of these bacteria, like how they reproduce and what are the main advantages or the diseases that are caused by the bacteria. So I think you are clear with this. So any doubts? Okay, then tomorrow we'll see about the next kingdom, that is Kingdom Protista.